Good afternoon. Good afternoon, everyone.
Good afternoon.
Wherever you might be, and welcome to our webinar. And today we have the great honor to host Professor Michel GHANEM for a very interesting seminar on drought and the ways to select for it, to understand its physiology, especially that it will be a major stress for the crops in the coming years, if not decades. Professor Michel Edmond GHANEM is an agronomist and plant ecophysiologist who holds an agronomy engineering degree and a PhD in plant biology from the University of Louvain, Université Catholique de Louvain in Belgium. And he also holds an accreditation to supervise research, habilitation à diriger des recherches, from the University of Montpellier in France. Before joining Mohammed VI Polytechnic University as an associate professor of plant sciences, he was the program leader for genetic resources at the Land Resources Division of the Pacific Community in the Fiji Island, where he led the one and only Pacific International Gene Bank. Okay? Professor GHANEM has also served as at the International Center for Agricultural Research in the Dry Areas, ICARDA, in Rabat, Morocco, as a crop physiologist, where he has established and led the crop physiology laboratory. Prior to joining ICARDA, he was a lecturer of plant physiology at the University of Namur in Belgium, and he also worked as a research fellow at the CBAS CSIC at Murcia, Spain. I don't know what that is.
Murcia is a town.
Yeah, Murcia is the town in Spain, but the other
CSIC,
abbreviation, Center of
CSIC
Estudios in Spanish. Yes, it must be in Spanish.
It's the National Research Council in Spain.
Okay, very good. So Professor GHANEM is also expert at the Biosafety Advisory Council of Belgium on transgenic plants, and have served on the executive board of the experts working group of wheat phenotyping within the Wheat Initiative. Within the CGIAR research programs, CRP, on grain, legumes, and wheat, he served on board of the research management committees as coordinator of the work on product line coordinator and flagship leader on trait identification abiotic stresses. His research expertise covers plant abiotic stress physiology, genetic resources, plant modeling, and plant phenotyping. So we'll have the pleasure to listen to him talking to us about a very important subject on drought reality. What are the realistic physiological options to increase crop yield under drought? And that's a huge question by itself. So, Michel, my friend and dear colleague, the floor is yours.
Thank you, Professor Boulich, for the introduction. I'm humbled by what you said. I'll be sharing my screen. So bear a few seconds with me before I can. Okay, here we go. So is the screen okay? Is that all right?
Yes, it's okay. Yes.
Okay, very good. Okay, so thank you for the for the introduction. As I said, I'll be discussing today some realistic physiological options to increase grain yield under drought. And I must say at the very beginning that this, what I will be presenting here is a joint work by Professor Tom Sinclair and myself that is submitted to a journal and is currently under review. And I'll say a bit why we decided to write that paper, because of many things we have seen lately about options to increase yield under drought. So I just wanted to start with a bit of context. Today, I don't know if you know, is the Fascination Plant Day. Every 18th of May, there is a series of networks all over the globe of actions that are taking place. So maybe Professor Boulif in ESAFE next year, we would think of organizing something on the 18th of May to show that really plants are fascinating. This is what actually has been my bread and butter for the last 15 years when I started studying those fascinating organisms. So of course, drought is something that you hear about. This is something that I took from The Guardian last week, and you can see that Madagascar is experiencing worst drought in 40 years, which is endangering millions of people's lives and food security in Madagascar. But this is something that you could also see in on more scientific type of studies. This is the US Drought Monitor. As you see, it was issued again last week. And you see that substantive amounts of California and other places are submitted to an exceptional drought. So this is something that is going to face the world where we live more and more as we go, especially under different climate change scenarios. So what do we do against that? First thing that comes to anybody's mind is that plants, of course, need water to grow. This is a global truth. And you ask a kid and he will answer you this question. So the first thing to do is irrigate. And we didn't invent that. I invite you to next time when you're on your way between Marrakesh and Ben Guerir, to stop at Sidi Bou Othman and visit those fascinating reservoirs that I'm showing here. That were built in the 12th century by the Almohad, the dynasty. And Morocco is also known for a long tradition in irrigation and in ingenious irrigation systems like those Khatara air shafts that you would see. You just go on Google Maps all around the house plain and Marrakesh, and you will see them. And even within the city of Marrakesh, you would be able to see them. So this is, I took a Google map of the Les Jardins de l'Agdal. So the basin is somewhere here. And as you can see, I don't know if you can see it properly, but those are the air shafts here that have been incorporated within courts. I mean, tennis courts, obviously, and football courts. They're still there, and they feed the basin there. So this is something, irrigation is something quite old. And Moroccans have been heavily involved ingeniously, I would say, in looking at several options. But recently, we started looking at other options that tweak the plants. Well, this is, again, nothing so new, but in the recent, I would say, 100 years, it was extensively studied, especially by plant breeders, with all the GMOs approach that is available to us, like this Monsanto Drought Guard corn. So of course, yeah, tweaking the plant is good, but let's talk before starting blindly to tweak the plants. And I posted here something that I shared recently on LinkedIn by my friend Lázaro Perez in Brazil. He's a very good friend of mine, and he shows actually that the breeding for those nice yellow tomatoes could have been done in a different way that gives more nutritious substances only if the plant breeders could talk to physiologists or other people, maybe nutritionists, rather than just looking at the genes, because we had several options. So I could have written that post because this is something that I constantly advocate for, multidisciplinarity and talking. Why it's important to talk? It's important to talk so we can avoid this kind of situations. That's a painting that I also cherish. It's Saint Anthony preaching to the fish. So we would avoid talking to each other, breeders, physiologists, agronomists, and other people, and not understanding each other at the end of the day, while we share, of course, common objectives. So that's why I think it was a good idea to set the scene back about some simple considerations about plants and water, the relationships between yield and water. So I would start with a very seminal observations on the relationships between yield and water. I'll then speak about some of the models, the phenomenological model by John Pasura, the mechanistic description of water use, and some of the challenges of different perspective, what I call the intuitive perspective, and the other one that we hear a lot about is the gene modification perspective. And I will finish by giving some realistic physiological options. And that's just our point of view. Of course, bear with me, I'm being on purpose provocative. So my idea is to challenge all of these things, ring some bells, and raise red flags when they are needed. I don't pretend to hold the truth, nobody does, but this is at least my point of view, because as I said, I believe, and that's a quote from my mentor and a very good friend, Tom Sinclair, physiology, this is really where the fun is. And I do believe that this is what brought me actually into the physiological field. So, okay, let's do some history, because I think it's what the students nowadays don't do much, is looking at historical things. To look at the very seminal observations, like by John Woodward in the 17th century. This man, this good-looking man, he has designed an elegant set of experiments to demonstrate that water passed out of the plant leaves through little pores, and it's extremely astonishing the kind of conclusions that he have drawn, and I've copied one of them here. So observations of the qualitative relationships between plant growth and transpiration reached a peak around 100 years ago, specifically by the work of two scientists, Briggs and Chance. So this is Briggs here. And if you look carefully at the pictures of the experiments that they have done in the 20th century, 1913, 1914, those are lysimeters. For those who know my work, I know that they probably know that I use extensively lysimeters in my experimental approach. So basically, 100 years ago, we're still using the same kind of tools, adding more things here and there, but nothing that substantial different. So the results of Briggs and Chance, they indicated that there is a qualitatively close relationship between growth and water loss. Later on, this brilliant man, Dewitt, he's a Dutch famous scientist. In 1955, he presented an analysis based on a data collection that was collected in the early part of the 20th century. And he found that by plotting growth, so this is growth here on the y-axis, against transpiration, and that's an important thing, divided by an index of atmospheric humidity, that there is a single linear relationship between those two variables for each of the species. So you see here sorghum, wheat, alfalfa. So Dewitt pointed out, he took actually lots of data from different cultivars, different environments, different soil texture, soil fertility, and importantly, water regime. And he showed unambiguously that plants, of course, need water to grow. And unfortunately, he didn't give much indications about the possibilities of altering the relationships within each and every species. So you can see it's fairly constant. What changes really here is the slope in each of these different graphs. And this is something to remember because that is another thing that we will be pointing at later on. So he didn't give much indications for possibilities of improving, but since it was equally important to look and produce in drought-prone areas, there was a description in the 70s by, again, this famous Australian scientist, John Pasura, that shows that the dependence of crop yield, which is expressed here by this equation, that is dependent on transpiration times harvest index, which is the ratio of the grain yield to the crop total mass. And then importantly here, a factor here that is called water use efficiency. Personally, I have issues with the water use efficiency definition. But this phenomenological model of John Pasura it's important because, again, it linked yield to transpiration. The problem is that it didn't give much insight about, it's not a mechanistic equation, and that's why constantly we hear that, okay, so we want to increase yield, you just have to increase the water use efficiency. Yeah, well, that's good, but how do you concretely, I mean, in practical terms, you increase the water use efficiency? So that's not a mechanistic equation, and that this is not an equation that takes into account the dominating influence of atmospheric humidity on the relationships. If I go back to the graphs by Dewitt, you can see that he divided that by an index of atmospheric humidity. So the model of Pasura has its merits, of course, from an agronomic point of view, it has its merits, but it's not a mechanistic one. So it doesn't tell us much on how we can improve that. And although the empirical approach of Dewitt, the phenomenological description of Pasura, they've all, as I said, added perspective on drought limitation to yield. None of them, unfortunately, offered a fundamental description on the variables that are resulting in the yield limitation under drought. So that was done by two scientists, one of them that I know very well, that is Professor Tom Sinclair, who published a very seminal and important paper in 1983. I was 5 years old at that time. And I really invite all the students to go back to these old studies because this is extremely important. So he offered an expression based on derivation that describes the gas exchange by crop canopy, and the derivation started with instantaneous expression of leaf gas exchange leading to a description of yield as a function of transpiration. And as you can see here, the above equation is important because it's an integral, so it takes time into account, which is dt here, and it takes it into account, and it gives some insights about possibilities of changes. And we're going back to this equation, so I invite you to keep it in mind. It also integrates the vapor pressure deficit, which is the atmospheric humidity that was mentioned earlier by Dewitt in his derivation. So the above equation is of course consistent with the previous expression that yield is intimately dependent on the amount of water available to support transpiration. This is something we need to remember. So specifically, yield cannot, unfortunately, exceed the amount of water that is available to a crop to support gas exchange. So if a drought develops, yield ultimately can no greater be allowed by the available water. Unfortunately, most of the I would say those who try to improve that ignore this physical reality and overlook this physical reality in some kind of a wishful thinking about increasing yield under water limiting conditions. So I'll go back to this equation, keep it in mind for later on. So unfortunately, when people started in the last 50 years or so in looking for solutions for drought, they ignored many of these approaches, the empirical of Dewitt, the phenomenological one of Pasura, and of course, the mechanistic description of Tanner and Sinclair. And they started in exploring what I call intuitive approaches. So things that ignore most probably the critical realities of grain production, but seems of some kind of a common sense. The first one is crop survival. Okay, so crop survival is something that you often see in many publications, specifically those that are related to changes in specific genes. So we look at drying substantially the soil. We submit the plant to a very harsh type of stress, and we look on the plants that have survived, and we take records like this one, take records of the one that we've re-watered. So this is the transgenic ones, and we can see that they're surviving, but they're ultimately not producing much than the wild type ones. So this is of course of a little practical benefit for farmers, definitely. I mean, farmers don't eat bonsais. We need grains. We need something that is produced by the plant. So if yield is not there, even if the plant survived, this is not feasible. And the recovery from the survival-threatening drought is an economic disaster for the farmer. Even if you recover and you get some growing crops, you will not be able to commercialize, and therefore it's still an economic disaster. So definitely crop survival, as we often see in publications, is definitely not relevant for crops, the food crops. The second one on the intuitive perspective is the osmotic adjustment. So this goes by simple hypothesis that accumulating constitutively osmotic compounds will allow water to go inside and sustain the activity. So, and this is what is explained here in the graph that I took from a publication by Neil Turner in 2018. So that seems like of a common sense, of course. But if you look at the series of the publications that, depending on species, etc., and that was compiled by Rashid Seraj and Tom Sinclair in 2002, and you see that the advantages are not always clear-cut, as usually we tend to present. You see, sometimes it's yes, no for barley, no for maize, rice, cotton, we don't know, it depends, only when severe drought is stressed. So it's not very much straightforward. And I would like to give you a few ideas about why this is not always the case. So there was a nicely designed experiment by Amber Besseley in 2019, who took actually two maize lines, one with a greater osmotic accumulation and the other one without osmotic accumulation, and those are near isolines. So basically, there are no noise. I would say no genetic noise behind that. And you can see that the extraction, the one with the greater osmotic accumulation, initially extracted soil. So you see here the soil extraction is much higher at the very beginning than the one that did not have the osmotic accumulation, which is fairly okay. I mean, that's good, consistent with the hypothesis. But the problem is that if you sustain that at the very beginning, the more rapid decrease in soil water content you have, because you're pumping water, resulted on an early date of exhaustion of the soil and for a decrease in the soil and the plant physiological activity. So the difference between those two lines is that the one with greater osmotic accumulation extracted water in the beginning quite more than the other one, but ended up not sustaining itself because it depleted the entire water. So the merits of an osmotic accumulation in the field are also dependent on the seasonal dynamics of water availability and its use. If you would have water that is available throughout the cycle, or you can reach it, then why not? If you don't, you will be depleting the water and not sustaining your physiological activity on the long run. The second thing is that the total water, if you calculate a bit, the total water extracted from the soil, it's virtually really unchanged. That's not much for osmotic adjustment due to really very small increases in extractable volumetric soil water content at very low soil water potential. And in some scenarios, the maintenance of a turgor by osmotic accumulation, as I said, as water deficit develops, is likely to give us results that are just the opposite of what we think is beneficial to the crop. So osmotic accumulation, main message is, let's be careful. It depends on the scenario. The other one that is repeatedly coming back and forth is the root extension, the rate and depth of root extension. And actually, the problem of root extension and the rate and depth of root extension is more or less the same as the confounding the response by the osmotic accumulation. So certainly, of course, if you increase root extension into a wet soil, that could be advantageous for drought conditions. No one can doubt that. However, if you have plants that have greater capability for deeper rooting, this might prove, again, non-useful under field condition. I insist a lot. Why? For sometimes if you have chemical or physical barriers that prevent the plant from going deeper or having a deeper root extension. So once you reach that soil barrier, the acquisition of crops by the plants is limited to the original water storage that is in the soil that is above this barrier, and it's not always beneficial to the plant. The second point that I wanted to make is that the increased root proliferation does not necessarily relate to more water extraction, because the water uptake is increased a little once modest root length density is reached. As you can see on two different studies here, in 1994 and in 2005, you would, in two different conditions, one in the field and the other one is in controlled conditions in small pots. So you can see that at a root length density that is beyond 2 centimeters, the amount of extractable water is virtually unchanged. Likewise, beyond 0.3, and I said the difference in the values is due mainly to the nature of the experiment itself. One is in the field and the other one is in controlled conditions. So again, the soil water content is not unchanged after a certain level, which is quite small. So there is no need It doesn't mean if you have more roots that you will be extracting more water. And that is something that has been shown by, there is a review by Vadez in 2014 that shows that. So I invite you to read that too. So the other thing that is, sometimes there is a negative aspect of rapid root extension, is that, as I said earlier, of root osmotic adjustment, is the great risk of early seasonal depletion of water that actually results in increased vulnerability to drought. So this is here, what you can see here is a simulation study of acquiring more of a root extension, root depth extension. And as you can see here, this is the probability of having either a grain yield loss or yield gains. And you can see that in most of the environments, this is the Western United States, and it's for soybean. And you can see that there are in most of the cases, there are negative effects on yield. Except in some situation when the soil is constantly being replenished. So again, root depth and rate and root extension is not a magic bullet. It depends heavily on the context that you're seeing. There's another and intuitive approach which says, okay, the problem is that seed set and growth are particularly sensitive processes to grain yield production. So let's sustain seed set and seed growth. Yes, yields are commonly decreased substantially when droughts occurs during the reproductive development. However, the direct impact of water deficit on reproductive growth is better visualized by the ratio of the grain yield to the total crop mass, which is the harvest index. So as you can see here, the harvest index is more or less fairly stable until the impact of drought on total crop growth is really extremely substantial. As you can see here in this study, I guess this is not yet published, but that's a study coming from Tom's group. In maize, there has been found that there's a very close relationship between the decrease in seed mass and the decrease in total crop mass with the water deficit, as you can see here. And because it's the correlation is 1.76, you would see that the major decreases in the harvest index did not occur until you reach a crop mass, total crop mass that is decreased to less than 1,100 grams. So it is really until it is very advanced that the impact could be seen. So again, this is something that is not a silver bullet. Okay, so recently, and this is this paper is one of the reasons that prompted us to write this study, is specifically this paper that appeared in 2020 in Science that says in the title, "The Physiology of Plant Response to Drought." And if you dig a bit into what's written in the paper, it's basically molecular analysis on Arabidopsis and again, hormone signaling, specific cells, cellular domains, etc., etc. Not criticizing this work, because just to give you an idea, I started my PhD working on those hormones, specifically on cytokinins. This was my PhD, and abscisic acid when I was in Spain, and the interaction between the two, specifically on salt stress, salinity rather than drought. But so there's a common thing that is what we call the gene modification perspective, that says, okay, let's modify the gene. And let's look at studies that correlate gene expression and identify a few of them and say, well, let's go and change one of them. It's exactly like like this picture, where it's extremely difficult to distinguish between correlation and causation. It's not because this cat is sitting in here that he has caused the degradation of the concrete. So let's be careful and distinguish a bit the correlation versus the causation. So what do I think of these gene modification perspective? Again, this paper examines plenty of basically phytohormones, abscisic acid, brassinosteroids, transcription factors like the worky ones, auxins, etc., etc. And it goes on engineering individual networks and components or single genes. The problem is that many of those genes are upregulated. When they are upregulated, they turn to have a negative growth trade-off because it's extremely complicated network. If you change one of the network's key players, you might end up messing with the entire thing. The target is extremely complex, and the underlying molecular networks are also extremely complex. And most of the studies, if you examine most of the studies that are published on these fantastic hormones, are in non-crop species, and basically this plant. We actually, when I started my PhD, I started working on this plant, and it was abandoned after a few years. It had its merits, Arabidopsis, because it gives us lots of insights about some of the molecular mechanisms and genes, etc. But it's not a crop, so you will have to work on really non-crop species, on really crop species to be quite relevant. And in most cases, if most of these studies, they have a strange experimental setup, which goes for either taking out very brutally the plants and leaving them for 2 hours on the bench, and then measuring those who have survived and those who haven't survived, or imposing a very harsh water deficit and saying, well, those who have survived, again, drought survival that I have discussed earlier. The other problem is that this approach overlooks two essential issues in my point of view. One is the scale of biological organization. Means if you cross between the gene that you modify to the crop itself, there's a lots of intermediate levels. You come by pathways, cells, organs, and then a plant and a crop. So the further you are between those two different elements, the more difficult is to establish really kind of correlation, because you're missing lots of intermediate building blocks. The second thing that these studies overlook is the temporal context of drought in the field. So there is no such thing as a single drought that happens once in every location. Every year it's exactly the same. Every year it's more or less the same type of drought. Even if we can identify type of major stress patterns, you still have to look in a certain number of years to have something significant. So the temporal context of drought in the field, again, in the field, not in pots, is completely not taken into account, because, I mean, basically they test them under controlled conditions. And they don't take into account that a modification here might be useful in one place, but a disaster in another place. So there is, as I said formerly, there is no such thing as a universal drought scenario. Even for a certain location, there's a suite of water deficit scenarios that vary in intensity and timing, and that are modified by so many aspects, like characteristics of the soil, the weather, and the type of management that we impose on the plants. And there's a very important also paper by François Tardieu that says really the most relevant question on drought tolerance is probably does a given allele or a trait confer drought tolerance, but rather does a given allele confer a positive effect on yield and an appreciable proportion of years and scenarios in given areas. And that's why it's equally important to do simulation studies, because it allows you to grow virtually plants in your computer, but examine a certain number of years, 30 years, 50 years of climate scenarios and above, and see what's happening there. So that's a tool that we should be more and more using to, and I'll show later on a few examples about that. So after demolishing all sorts of things, you must certainly be depressed and saying, okay, though there is nothing much we can do. Let's go back home and stop treating drought. Well, it's true and not true. There are still some realistic physiological options that shows that there's a consistency between the empirical analysis that I've mentioned earlier in the introduction, the description by John Basura, the mechanistic derivation by Tom Sinclair. That, as I said, is crop yield is ultimately constrained by the amount of water that is available to the plants during the growing season. It means that this constrained yield possibilities is an important reminder that any plant modification without a major increase in water availability, and I insist on that, can only be expected to result in very small or at best moderate yield increases. So that's why the realistic view of yield sensitivity to water availability indicates that we still have some tweaks that are more or less realistic that we can do. And for that, I want you to go back to this equation that I have put in the very beginning. So that comes from the derivation of Tanner and Sinclair. And again, it takes into account aspects, the K, which is the slope of the graph, if you remember, that was established by Dewitt, which is very consistent, transpiration and VPD, and of course time, because there is an integral. So it allows to take the temporal and dynamic aspects into account. So what are the options here? And we've actually, after long reflection with Tom, we came out to 4 of them. 4 of them, and depending on the likeliness, we've decided to put the arrow a bit bolder, or so we will. I'll start by this one, the increase in this K, which is not very likely. Sustain the harvest index, increase the available water, and decrease the effective VPD. Okay, so I'll start by this one. The first one is increasing K, and K is actually the transpiration efficiency. It's the slope of the curve, if you remember, the 3 graphs that I've shown by Dewitt. The slope of this correlation is the transpiration efficiency. And if you look at the analysis that comes from Tanner and Sinclair in 1983, gives a bit of mechanistic understanding about this value. So this value has a formula. This is the formula of the transpiration efficiency of this K, and actually it depends on 2 main things, B and C. B is actually the photosynthate conversion to plant mass, carbohydrate, protein, or lipid content. So how do you convert the photosynthate to carbohydrate or to protein and lipid contents? And it depends on the type of crops that you're growing. Some crops are grown for their lipid content, some other are for their protein, and some others are for their carbohydrate. And there's the C term, which is basically depends on the differences between the internal and the external CO2 concentration. And if I don't want to dwell on this equation, if you would like to see more about how is this happening, I invite you to look at our paper in 2020 that we published in Crop Science, and in which we look at the plant-based prediction of canopy transpiration instead of the meteorological approximation. But more or less, what it says that this slope of efficiency is higher for crops that are of higher energy content, like maize, sugarcane, a bit lower, intermediate for wheat and rice, and soybean and peanut. So it's actually depending a lot on energy, protein content, or lipid concentration. So first thing is, if I go back to the equation here, to the initial equation, if you manage to increase K, you would be able to increase the. So how do you increase K practically? Remember, here the equation depends on two things, on the photosynthetic conversations and the internal versus external CO2 concentration. So how do increase yield? First thing is to go for low energy grain. So either you go for breed for low protein or lipid concentration. That would definitely increase the K value. But as you probably know, crop species are grown for their unique biochemical composition. So wheat is grown, for instance, for specific protein concentrations, if you want to use it in baking. Soybean is grown for the value of its high protein and high lipid content. So the market imposes penalties if the biochemical composition is different from the prescribed concentration. So that's an option, but it's not really likely because of market issues. The other component of K is that the photosynthetic characteristics of the leaves. Certainly, if a C3 photosynthesis species could be transformed into C4, that would be a major increase in K. Again, the problem is that this is a complete makeover of the plant's photosynthetic system and remains like a monumental challenge. I mean, I would love it to be possible, but I don't think it would be feasible because it imposes a monumental challenge in the whole plant photosynthetic system. It's not as easy as it seems. So those are two unlikely options in our point of view. So the other option is, again, looking at the CO2 gradient. So if you tweak the CO2 gradient to achieve an increase from the atmospheric to the leaf interior, you would increase K by, so if you, one approach is to decrease stomatal conductance, which is, this is a one way to achieve the gradient in CO2. That's a variant of what we call the slow growth approach, in that the overall photosynthesis rate would be decreased, so the carbon accumulation would be decreased even when water is not limiting. But that's, again, it offers solutions, but I don't think it offers a major improvement because, again, a slow growth approach might not give us much yield. And as I said, we need to be looking more into increasing yield because this is what we are looking for at the end of the day. So the second one is sustaining the harvest index. Remember what I said, that the crop yields are often thought to especially be sensitive to drought during anthesis, which is completely true. The yields are commonly increased with drought during this sensitive period of plant development. However, we need to bear in mind that the critical variable in assessing the impact of drought during anthesis is harvest index. Remember, harvest index, not absolute yield. If the harvest index is unchanged or only slightly decreased, which is most often is the case under water deficit, the impact of drought on its crop accumulation is limited by the amount of water as defined by transpiration, not uniquely reproductive development. So the critical variable to measure is simply harvest index in response to drought, which can be done effectively by just harvesting few individual plants. And only if harvest index has decreased substantially in response to drought, remember the graph that I've shown, that can yield be improved by overcoming the negative response on early physiological reproductive stage to drought. So that's a likely possibility, but again, not easy to put in place. The third one is increase the available water. This will have an impact on increasing the potential of crop growth. As I mentioned earlier, the yield is dependent, highly dependent on the available water. If you want more yield, you would need more water. So in addition, of course, to management options, because those are something that is not discussed here. I'm not discussing the things that are related more to agronomy that can achieve a lot advancement, like irrigation management, conservation agriculture, mulching, and so on and so forth. Those are management options that would lead to an increase T. If we try to match sowing date and crop ontogeny with rainfall patterns, so we don't fall the crop cycle within the drought, that could be something extremely useful. Of course, osmotic adjustment, as I mentioned earlier, is something that could be taken into account, but I think this will result in very small shifts, as I discussed earlier, to increase the accessible volumetric water. The routing, it's still it's still a viable option, but we need to take into account that water in the deep soil layers need to be recharged by off-season rain, so that the water actually exists. It doesn't make sense for you, if you were a plant, to grow more roots if you won't be able to find water in the deeper soil. And there must be, of course, no barrier, as I mentioned earlier, for the deeper rooting. And the temporal dynamics of water use need also to be regulated, so that the rapid root extension does not result in an early season water extraction, and therefore vegetative during the vegetative development, causing the crop to be even more vulnerable to the late season drought. Remember, it's like having it's like racing a Formula One. If you want to reach out the finish line, you can choose two options. Either you spend most of your fuel at the very beginning, and you are, of course, you would be ranked first in the first or second round, or keep the fuel for when you really need it. It means when you want to really cross the finish line. In other words, when you want to have yield. So that's those are the considerations that we always need to keep in mind when we look for tweaks for osmotic adjustments, or even for rooting. So there's another one, that the fourth one, that we think could really be extremely interesting, is the one that conserves water by limiting transpiration under high VPD. You know, VPD is vapor pressure deficit, and as it says, it's the deficit between saturated moisture air and the actual air. So it's a deficit. The more it's higher, the less humidity you have, and the more water demand on plants. But from the outskirts, you tell me, well, VPD describes actually the status of atmospheric humidity. So there is absolutely nothing you can do about it. When on initial inspection, it seems that this is a given condition of the environment, and there is little opportunity to modify the value of the variable for drought improvement. However, we think that this is one of the prime candidates, candidate variables for increasing yield with limited available water. If you remember the equation that I showed by earlier, where you have an integral, the VPD is included as an integral in the equation that I have stated earlier. Let me go to it. Yeah, you see here, this is included in the integral. So it reflects the temporal dynamics of this variable. And the effective VPD of a day depends on the times of the day when the stomata are open and transpiration is essentially unimpeded. If the stomata remains fully open throughout the day, then the daily VPD, weighted VPD, is actually the VPD status of the air. There's nothing to do there, and it's effectively an atmospheric variable that we can't do much about it. That is only when the stomata is open the entire day. If plants regulate their stomata so that they are partially closed in some times of the day, let me show you an example. As you can see here, so this is a plot that shows the vapor pressure deficit. So when it goes up, it means that the air is getting drier. And this is the time of the day. And as you can see, during midday, this is the evolution of VPD during the day. It starts at a very low values early in the morning, goes up, and then midday reaches a certain maximum, and it goes down during the day. And that's the transpiration rate is more or less followed by follows that simple trend. It means that the transpiration rate is low at the very beginning in the morning, goes up during the day, and then goes down. Imagine if plants regulate their stomata, so there is a partial closure as stated here. So they are partially closed at some times of the day, usually during midday when the periods of VPD are the highest. And in that case, you would only decrease the daily weighted VPD in the equation that I have mentioned before. So the conservation of water by partial stomatal closure during this period of high VPD not only benefit late season physiological activity, because it's again something that you can keep your resources when you really want to use it, making small economies, everyday accumulating economies. Of course, you should not use it, lose it in another way. It increases globally the so-called water use efficiency. So the effective use of water is really increased. Another option is by looking into some of the governing parameters, like the low hydraulic conductance between the bundle sheet and the companion cells. This is the stomata and the companion cells. If you look at the hydraulic conductance of water loss, if you go for leaves with low hydraulic conductance under well-watered conditions, we could also change and decrease the VPD. So I'll show you a few examples that come from simulation studies. So those are studies on soybean in both Africa and the United States, and that shows actually a positive percent of more than 85% of grain weight increase in the drier situation, and a bit less the blue dots here and there. So that's a simulation study on soybean in Africa. And that's another simulation study in the United States, again on soybean, that shows a probability of having higher yields under specific circumstances, specifically when it's dry. So that's soybean. Of course, maize, similar results have been shown on maize, again by by the group of Tom Sinclair. We also did a simulation study on lentil, because when I worked for Ricarda, we have screened lentil lines for this VPD response. And we did a simulation study by Julie Guiguiton, who did that study during her training at Ricarda. And she had found that there are major yield benefits of a lentil line that restricts its transpiration on high VPD, and that those major yield benefits that you could see here, either on average grain yield increase or an increase in yield probability. And the greener, it means the more positive effect you have on yield. And if you can see, the effect is higher when the rainfall the rainfall is low. So the effect of these kind of response is more visible when it comes to dry lands. So that is something that we did on legumes. Just for me to say that not only our group did things that showed that this could have a potential. This is a study done by an Australian group, the group of Graham Hammer and Kain Chenu in Australia, totally independent from ours. And they actually show that for wheat, in New South Wales, Queensland, there are positive impacts on having wheat lines that have a restrictive transpiration over high vapor pressure deficit. So you don't say that this is only the Sinclair clan that claims that this thing is absolutely fantastic. Well, we also did another simulation study on a country that is closer to us in Morocco. We did that on wheat with the group of Walid Sadaf from Minnesota. And actually, the study was about simulating a line that has a restriction on VPD at 2 kilopascal, which is quite high vapor pressure deficit values. And if you can see, this was a study that has been done in a country that is close to our hearts too, Tunisia, close to to Morocco geographically. And you can see that there's a probability of yield increase, specifically in this area, the central area of Tunisia, much less in other places. And if you look why, if you look at what are these regions equivalent to, you can see that this is the driest area of Tunisia. I mean, not the driest in absolute terms, but in agricultural terms. Where you have drought, you would have certain benefit of having a wheat line that restricts its transpiration on high vapor pressure deficit. And I always wonder, how come that international centers working for the CGIAR haven't started a breeding program by looking specifically at these lines, which is something that is very, very easy to screen for. And we're not the only ones who did that. Pioneer, which is a company that sells and develops commercial cultivars, have done substantial work on maize hybrids. And those actually are maize hybrids that were sent with numbers to the Sinclair lab for testing their VPD response. And you can see that there is one that restricts its transpiration and has a breakpoint somewhere here at the VPD values around 2. So that's the transpiration rate plotted against VPD. And the other one continues to transpire as if nothing was happening. And it turns out that this interesting line is a commercial line that has been commercialized by Pioneer and branded as Aquamax. So you can see that this commercial line, Aquamax, is actually based on this specific trait. It's not just randomly selected. It has been selected based on this kind of trait. It's a non-GMO. It's based just on selection techniques, which is quite easy to do when it comes to the VPD response. Okay, so I'll conclude. I do believe that, I mean, we all know that plants need water to grow. And in all discussions of growth, a healthy reminder is needed that yields cannot be increased beyond what is really available in terms of water. So the empirical analysis of the wheat, the model of Pastura, and the fundamental derivation of Tanner and Sinclair, they are all anchored in water availability. It means transpiration. And ultimately, the increases in yield would require increased water input in the form or either precipitation or irrigation. But there are suggestions for yield increases that are offered here that we discussed that could result in tweaks, that could result in yield increase, but they can, I insist a lot on that the extent of any yield increase for any of these traits will depend on the cropping situation. Any trait could be a very good trait in a certain environment. You take it to another environment, and it's definitely a disaster. So the intuitive solutions that I have discussed earlier for yield improvement under yield conditions turn out not to be so helpful at the end of the day. Maybe in some cases, because opposite response is desired. There is one intuitive response that it seems, again, attractive, like the acquisition of deep soil water, but maybe it is the most difficult to implement. And as I said, discussed that, the challenges for roots. So the most consistent approach in our point of view is the water conservation traits. One of the major ones is the decreased VPD, and that these responses have been associated with increased sensitivity to soil drying. So before attempting to install these drought improvement traits or any other trait in specific crops, maybe it's good to start by using a mechanistic simulation model to explore the potential impact on yield, and then you can screen for it and you can select for it later on. With that, I thank you and I leave room for comments.
Thank you very much, Professor Ghanem, for this very insightful and very interesting presentation. Yeah, it is rather rich and implemented, well illustrated with with different graphs, and many people have enjoyed it. And because we are running out of time, I shall point out few questions to you for you to answer. Let me start by this one from Mr. Omar Idrisi, saying in many regions drought episodes are most of time occurring with heat stress. Are the realistic physiological options you suggested to increase yield okay for such scenarios? Thanks.
Yeah. So yeah, thank you. Very interesting question. Exactly. There is often a confounding effect of I would say temperature. I would like to use more than temperature kind of stress. And actually, if you look at temperature stress, there have been studies that were published by Lobel in 2014, 2017, etc., that shows that what we call the heat stress effect is more exercised through a VPD effect, because if you have a higher air temperature, the VPD will go even higher, so the evaporative demand will go higher. So of course you have an effect of the temperature per se on the plant development, but there is an indirect effect on VPD. And definitely the VPD approach is one thing to take into account, bearing in mind that if you were a plant, you will have the choice to make two options. Either you have to cool yourself and evaporate, lose water, or shut your stomata and heat eventually the leaf surface, which is not always a good balance to find. But definitely the vapor pressure deficit response is one of the things that could be looked at when looking at heat stress, temperature stress.
Okay, thank you very much. Here is another question from Mr. Diop Masamba, our friends from ITEC. He says, hello, many thanks. What is the potential effect of mycorrhization in plant root extension in drought periods?
Well, yes, mycorrhization would be something that to look at. Again, bearing in mind that it could be beneficial in certain environments if it allows the plant to have access to more water, if it allows the plant to deal with the stress in a different way. Unfortunately, the studies that I have presented, they're studies that were performed in controlled conditions, and I don't think they have looked into mycorrhization effect in plant roots. But I guess this is something to look at again from a scenario perspective, because I do firmly believe that there is no magic bullet, but there are solutions that depend on the context heavily.
Yes. Okay. The connection went frozen for some time.
Oh, I don't know if you missed my answer, or should I repeat the
Yes,
answer?
we missed the last part of it, but that's fine.
Okay. Okay, that's good.
Okay, let's go to another question, and this time by our yes, you're being popular this year, this time today. Well, we're glad that they are here. So he says breeders and the physiologists tried to keep high yielding potential under drought conditions, but when it comes to nutritional quality, there is no less physiological studies combining drought and nutritional quality. I guess you have understood.
Yes, yeah, no, I understood. I happen to know mice quite well to understand the question. I think mice is absolutely right. Focusing only on yield increase is certainly one thing to take into account, because the amount of calories that needs to be produced and the amount of yield production. But definitely looking at the nutritional quality is at the basis nowadays of any improvement. You could improve yield, but if the nutritional quality goes down, there's absolutely no point of doing so. So I guess he's absolutely right. There are not many physiological studies combining drought and nutritional quality, and that's why I'm more than happy to work very closely with my friend mice on this aspect.
Okay, thank you very much. It is our friend Omar Idrisi coming back with another question. What are key traits linked with the transpiration efficiency? High harvest index and low VPD that should be targeted when screening for drought tolerance.
Okay, yeah, so I agree with, I think I've explained that high harvest index should be something to target only when the effect of drought on harvest index is extreme at the extremes, is higher and higher. I think a low-hanging fruit is a VPD restriction approach that could be easily screened. We have developed screening methodologies for these things. You can easily look at the genetic diversity that are at hand for those who restrict their transpiration when it comes to high vapor pressure deficit values. And I think that's an easy way of doing so, rather than looking at high harvest index. If I had to choose between the two, I would go for restricted transpiration on high VPD, again, depending on the circumstances and the drought scenario that you're facing. Remember, on the maps that I've shown, in some places it was very good, it was very green, and some places it was red and orange. So not always magic bullet. There is no magic bullet in physiology.
Okay, here is another question by one of our students, William Makaza. He says the rate and depth of root extension. So do we have a specific ideal crop idiotype required to promote water use efficiency and mitigate drought in food crops?
Okay, first I have to insist on the difference between idiotypes and idiot types, which is often confounding, because I mean, an idiotype again is something that is quite depending on the scenario. We cannot take root extension as a universal trait that works everywhere and in all occasions. I don't think there is such thing as a specific ideal crop idiotype when it comes to rate and the depth of fruit extension. It depends on the scenario of drought that we are dealing with. Ideally, if you had water in the underground and you can access it, then why not? Let's go for a deeper root, provided that this deeper root, that this soil at depth is replenished once the season is over. Otherwise, if you cultivate the next year, there will be no water to harvest from. So I'm afraid there is no such thing as one idiotype. There are different idiotypes that are dependent on the drought scenario that we are facing.
Okay, thank you. So the questions keep coming up. So here is one from Ms. Salma Rwishi saying, very good presentation, thank you. I wanted to know your opinion about genetic diversity and genetics as whole in improving the response to drought stress.
Well, genetic diversity is key, because this is where we start looking at. I'm a big advocate of using the genetic diversity that we have in looking for some of those lost traits that we have lost during breeding. Our group, and when I used to work in Canada, looked at a domestication series of durum wheat by looking at the wild ancestors all the way to modern durum wheat varieties, French modern durum wheat varieties, looking at some of these traits that were lost. And we've discovered that actually during the breeding, some of the traits that are related to water conservation have been lost because we were breeding in environments that had high water availability. So tapping into diversity is something that we always need to look at. Genetics, don't get me wrong, I'm not saying that genetics or genomics are not very good tools. They're really good if you know what you're looking at, and if you don't forget the basic physics behind crop use of water. I mean, there are some immutable physical principles that you can't avoid when you want to, you can tweak all the genes you want if you don't keep in mind the basic physical equation of if you want to produce more, you want to have more water available. And eventually what's restricting yield is what I call is, well, sky is the limit, because it's limited by precipitation, it's limited by nitrogen, which eventually comes from the sky. And it's limited by by radiation, solar radiation. So genetics are good. They help us understanding the underlying mechanisms, but they're not a universal answer. And if we don't understand the physiology behind that, we can improve that genetically, but we won't improve anything, because when we take them to the field, they will be completely useless.
Okay, well, thank you very much. There is another question from a friend of ours, Professor Manal Mhada. Low vapor pressure deficit affects plant nutrition. And she's asking, is VPD directly affected by plant nutrients?
As far as I know, so I don't know if the response, if Manal means the response to high vapor pressure deficit is affected by plant nutrition. I guess there are a few studies that have looked into phosphorus effect in West Africa on sorghum. But we still don't know the mechanisms, and definitely the question is extremely interesting, because I think the VPD response is affected somehow by plant nutrients. I expect it more to be influenced by things like aquaporins and genes that regulate the water hydraulics, I would say. But there were studies that show that specifically in phosphorus, there is an effect on the response. So definitely that is something to look for in the future. Okay.
One last question from our friend Dr. Rashid Marabat, the conservation agricultural specialist. And yes, he says, very thoughtful presentation on drought versus aridity. Did you relate greenhouse gas induced radiative warming to VPD? Did you relate carbon accumulation to VPD and hence to water use efficiency?
Yes, thank you, Dr. Mrabit, for the interesting question. There are some studies that look at the climate change and VPD effect. I actually pointed out to the studies by Lobel with different climate change scenarios. One thing that is unfortunately not enough taken into account is the changes in CO2, because that could have high impacts on the global the global response in climate change scenarios. So there is the second question related to carbon accumulation and VPD. Of course, when you restrict transpiration to high vapor pressure deficit, you might have small penalties because you're restricting, I mean, CO2 and water share exactly the same route. And that's also physics. So there would be some penalties on carbon accumulation, photosynthesis, I guess, but it could be compensated because the plants are able to, if we assume that the plants are able to sustain their growth for a longer period, then we can assume that those small negative impacts could be reduced on the long run. I don't know if I answered
the last question, but here is one that popped up just now from Aziza Zarruq. What about introgressing drought tolerance traits from crop wild species adapted to extreme environments into cultivated plant species? Can we consider it as one of the realistic options?
Yes, thank you, Aziza, for the very interesting question. I did not say in my presentation that doing introgression or breeding is not a realistic option. And definitely looking at crop wild species is a very viable option, because they would, as I mentioned earlier, contain things that maybe we have dropped during extensive breeding. So that's definitely a strategy, but you need to know what you're introducing. If we just take randomly one one wild relative that we think is drought tolerant and cross it with a cultivated relative, maybe we could have a different response. But if we know what we're looking at, it means if we do phenotyping for a specific trait, including in the crop wild relatives, then we would be able to do a more informed type of breeding scheme rather than just blindly introgressing lines saying, okay, well, Taoshai is interesting, so let's cross with wheat and we'll probably find something. We can't afford the numbers game anymore. Unfortunately, we can't afford having so many things at the same time. It needs to be hypothesis driven, including when it comes to wild relatives.
Okay, I hate to stop here, but there is another question from Mr. Ali Tiam about the mulching system. He says the mulching system is it effective in dry areas? With, of course, on beds, do not you think there is a problem of seed selection adapted to the soil structure of dry areas?
Sure. I mentioned actually that the management options are definitely something, and agronomy. I mean, we haven't explored agronomy as we should explore it. And I go back to what Dr. Rabbit was saying. I mean, he's a hero of conservation agriculture, and he knows that this is something that that could be eventually much more beneficial than any of those tweaks. So I guess anything that would reduce the water loss, so you keep it next to the plants, mulching is definitely a viable option for that. Whether we go for a seed selection that are adapted to soil structure, yes, that is something that is not always looked at, maybe except a few studies that were done in Australia, not on the soil structure per se, but on the sowing depths by looking at long coleoptile type of wheat by in CSIRO. So yeah, that's definitely one area of looking at soil structure. Yes, most of the time the soil has been completely left out of the story by those intuitive and genetic and gene-centered type of studies. And I guess it's time to put it back and go back a bit to agronomy.
Okay, here is another question from our colleague plant breeder, Dr. Amamu Ali. He says, what is the efficiency of breeding using physiological tools when we have an unpredictable climate? Drought is appearing randomly during the plant cycle, beginning, middle, or end of the cycle, he says.
Okay, thank you, Ali, and good to see your question. So you are absolutely right. And drought, as I said, is an unpredictable thing, and there is no such thing as universal drought. That's why it is important to go first for simulation studies, where you basically look at different many years. So you have a high probability of having an environment, having a typology of the environment that you're targeting for breeding. So I guess the first step would be using having a hypothesis in mind, using mechanistic crop growth models, and I insist on having mechanistic, physiologically informed crop growth models rather than just calibrated ones. So you would be informed of any probability of having yield increase in a specific environment by changing this trait, and then you can go and looking at screening for this particular trait. So I guess physiologically informed tools could be beneficial, but we have to take into consideration the location, the environment that we're targeting. You can go for environmental characterization of the most frequent type of drought that occurs in the 30 years or so. And you would find always ways of increasing the probability of yield increase rather than absolute values of yield increase. That's why, again, use crop models so that you are informed of the modification of a certain trait and then go for a selection for this particular trait if you think it's beneficial for this location.
Well, I think that was the last question, and I'd like to thank our professor, our speaker, our distinguished speaker for today, Professor Michel GHANEM, for a very interesting presentation that has sparked a number of questions from our viewers and followers, whom I thank very wholeheartedly, and thank you very much for keeping this great interest in our webinars and seminars. And we are doing it for you, and we're happy that you are taking advantage of it. Let me take also this opportunity to announce that the deadline for the applications of our master's degree, master's program in fertilizer science and technology is planned for May 31st. So please, if you know of candidates who are interested in this master's program, to start their application, to load their application on the platform, on the website, my.um6p.ma, or for more information to drop an email to the ESAFE@um6p.ma, or a message, a WhatsApp message to the number indicated on the screen, or simply visit the website of our school, asaf.um6p.ma. We thank again, and we thank you all, for all you followers, for your attention, appreciation, and your questions have enriched the debate. Okay, thank you very much.
Thank you so
and
much.
See you next week for the next webinar.
Thank you, Professor Bully.
Thank you, Michel.
Bye-bye. Bye-bye.